Uses of Protein Electrophoresis by Brandon Reid. The purpose of this experiment was to figure out what an unknown bacteria swamped my teacher's computer was. Some of the reasons that I did this project was because I was curious about the extensive steps medical researchers take to fight and eradicate disease, and as well as the steps afterwards that the medical researchers might take into deploying that vaccine into the public. My hypothesis was fairly easy to come by. I hypothesized that if I extract the proteins from various bacterial cultures, then I'll be able to visualize the banding pattern created through a protein electrophoresis and use this data to classify bacteria. Further, I'll identify and differentiate an unknown bacteria culture using the banding patterns of the known bacteria as a standard. During the first day of my lab experiment, there was a multitude of steps I had to do to prepare for the experiment. Some of these steps included pouring the agar into the Petri plates. Of course, you'd have to heat up the agar to then pour into the Petri plates. You kind of poured the agar into the plate so that it just kind of covered the bottom. It wasn't exactly an exact measurement. After you waited for the agar to solidify, you would then scrape more or less the bacteria from the slants that Evotech provided for me, and you would then take the scraped bacteria and then spread it across the agar plate, so more or less it was no part in the agar plate was missed, so you'd have to go back and forth repeatedly. After you're all done with that, you let the bacteria settle in for about 10 minutes or so, and then you put it in the incubator for an overnight warm-up. After you finish the first day, you would then retrieve your bacteria Petri plates, and you would start preparing different parts and for cutting the proteins, more or less. You would have to create the lysozyme. Following that, I was then prepare the bacterial lysates, doing standard amount of steps, including scraping the plates and resuspending the cells. You also had to remove the supernatant by centrifuging the protein. Day 3 is finally the day where you got to see your results. During day 3, you'd have to get the buffer ready to pour into the electrophoresis unit so that the buffer more or less protects the bacteria from being destroyed and makes it able to run better, per se. You'd have to load these gel cassettes with your bacteria lysate solution, and how you do that is you use a pipetter, and you load in about 8 ULs of the bacterial lysate into each gel. And you have to make sure that when you're loading these gels is that you do it slowly, because if you go too fast, your bacterial lysate solution might push in the gel too far, I guess, per se, and it would fall into the other wells. You just have to redo your whole experiment again almost. And that was actually what happened in one of my first trials, and so that is probably why I had to do multiple trials for this experiment. Then after you'd be done with that, you would then run your electrophoresis for about 85 minutes or so. It depends on how exact you want to be. I did 85 minutes, and then you would pull out your gel cassette and all of that. And then you would stain it with the Insta stain that Evotech then again provided for me, and then you would let it sit in this solution overnight that was prepared for me. This is the first gel with results. You can tell from wells 1, 2, 5, 6, and 10. They're all more or less identical. They all have 8 banding patterns. Their measurements actually on the acetate sheet, which I will discuss in the next slide, are 6, 9, 11, 14, 21, 27, 33, and 37. That's all in millimeters, and it's measured from the top of the well, which was overlaid by the acetate sheet. So you'd mark the acetate sheet, but you'd follow the well. So you mark it down from the well down onto each significantly being defined banding pattern, and you'd mark it all the way down. Then you just basically record your measurements in millimeters. As I was talking about before in the previous slide, the identical looking gels or wells are 1, 2, 5, 6, and 10, and they all have the same exact banding pattern measurement from the top of the well down. 6, 9, 11, 14, 21, 27, 33, and 37, and it was all in millimeters again. When I loaded this gel, this was not the order that I wanted. It was a different order, which we'll discuss in the discussion section of this PowerPoint. But the order for this went U, U, B, S, E, E, B, S, M, E. And those are all basically one-letter syllables for the actual bacteria names themselves. It was just shortening the word itself. This is the gel acetate sheet. It's basically just a clear plastic sheet of which I just laid over the gel after it was done with electrophoresis. So basically you lay the gel acetate sheet and line it up with the gel itself, and then you start marking with a Sharpie or any fine tip ink pen or marker you can use, and you measure it, or just mark basically the really definitive bands from the top of the well down. And if these gels look consistent, or the wells look consistent I should say, then you should probably try and find those same exact bands if those wells seem same by glance. This is the data table with all the measurements for each bacteria and which lane they were in. Once again, you can tell which bacteria is in which lane in their measurements. This is more of a definitive way of trying to identify the unknown bacteria because they're exact numbers and exact data. So you can see again that the unknown and the E. coli more or less match up with their measurements of 6, 9, 11, 14, 21, 27, 33, and 37, all once again in millimeters. This slide is more or less an overview of the data on the acetate sheet and the gel. It's more or less reviewing the idea that this E. coli and unknown bacteria are the same exact bacteria. Just talking about the measurements of 6, 9, 11, 14, 21, 27, 33, and 37. Those were the banding measurements. And you can tell from the beginning that just looking at the gel by glance that these wells, 1, 2, 5, 6, and 10, seemed pretty identical. This is the part where I more or less uncovered that E. coli was actually the unknown. This experiment was more or less of a success because I in the beginning didn't think I was going to actually figure out the unknown bacteria due to the failed trials in the beginning. 2 of which did not go well. There were 4 in total. Once again, I'm talking about some of the flaws I made during the experiment, such as one trial I didn't maybe cut the proteins right, and then another trial maybe enough supernatant wasn't destroyed, I guess. And then another trial I messed up the order, but that was actually the trial where I got the best results. Another trial, some of the wells had inconsistent amount of the liquid due to deformation in the wells themselves. Some of the wells were bigger than the others, which is really weird because I'm assuming it's manufactured. But now I'm just curious of what the medical researchers would now do. They have all this data. They know that this unknown bacteria, maybe an unknown pathogen, is actually E. coli. Would they then use an E. coli vaccine, or would they take further steps and then figure out how to treat this and treat the public? Thank you for your time, and I hope you enjoyed this PowerPoint that I put together for you today.